Hey everyone, this is Narendra and you're watching to my channel Bioguru. In this video, we are going to learn about pollen-pistil interaction. So now, all the events from pollen deposition on the stigma until pollen tube enters the ovule together referred to as pollen-pistil interaction. And pollen-pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. Pollination does not always ensure the transfer of compatible pollens, that is, right type pollens. Hence, the pistil has the ability to recognize the right type of pollen to promote post-pollination events. If the pollen is of wrong type, the pistil prevents pollen germination. This interaction is mediated by chemical components of the pollen pistil. The pollen tube reaches the ovary and enters the ovule through the micropyle. Then through the filiform apparatus, here you can see, through the filiform apparatus it reaches the synergids. In this way the pollen tube grows and thereby it releases its two male gametes. Now let us study about the artificial hybridization. Hybridization is a process of crossing two genetically different individuals to result in a third individual with a different, often preferred set of traits. It is a method to improve crop yield. In this method, it is essential to ensure that the right kinds of pollen grains are used and the stigma is protected from unwanted pollen grains. And it is achieved by two processes. One of that is emasculation. So what is emasculation? It is the removal of anther from the flower in bud condition. Right? So that is called as what? Emasculation. So here the anther is removed from the bud if the female parent bear bisexual flowers. Then next process is bagging. The emasculated flower is covered by a bag, preferably butter paper, so as not to allow contamination of the stigma by unwanted pollen grains. When the stigma of the bagged flower receptive, the collected pollen grains are dusted onto the stigma and then the flower is rebagged. If the female parent is unisexual, emasculation is not necessary. In this case, the female bud is directly bagged. When the stigma turns receptive, suitable pollen grains are dusted onto it so as to allow germination. Now let us study about double fertilization. In double fertilization, the pollen grains, when they fall on the stigma, pollen tube is germinated and it reaches to the ovary into the ovule through by micropyle region. And from there it enters into the one of the synergid and then it is going to release the two male gametes. So one male gamete fuses with the egg cell. So here there is a egg cell, right? And here there are two male gametes in the pollen tube. So after entering into the synergids, it is going to release both the male gametes. So in that, one of the male gamete is going to fuse with the egg cell, right? So what is going to happen? Let us see. So here after fusing with the egg cell, it is going to form a structure that is called as zygote and it is a diploid, right? And the fusion of male gamete with the egg cell is called as here syngamy, right? So there was one more male gamete and it is going to fuse with the two polar nuclei. So each nuclei here is haploid. So the male gamete is also haploid. So there are two polar nuclei already present and in that another one male gamete come and fuses with these two polar nuclei in order to form a triploid nucleus that is called as primary endosperm nucleus, and this is termed as triple fusion. Why triple fusion? It is because here two already nuclei are there, polar nuclei, to which one male gamete is going to come and fuse with it, right? So resulting into the a structure or the nucleus, it is called as primary endosperm nucleus, which is triploid. So now short form it is called as PEN, that is primary endosperm nucleus. So the fusion of male gamete with the polar nuclei is termed as what? Triple fusion. So resulting into the PEN. So one of the male gamete after fusing with the egg cell, it forms the zygote, and the next another one male gamete, when it fuses with the polar nuclei, it forms the primary endosperm nuclei. So since two kinds of fusion, that is syngamy and triple fusion, takes place, the process is known as double fertilization. And this double fertilization is the characteristics of flowering plants. After triple fusion, the central cell become primary endosperm cell. The primary endosperm nucleus give rise to the endosperm, while the zygote develops into the embryo, whereas the synergids and the antipodal cells, they get degenerated. Now let us study about post-fertilization events. So it includes formation of endosperm, development of embryo, development of seed and fruits. Let us understand one by one. First one, formation of endosperm. Endosperm develops before the embryo because the cells of endosperm provide nutrition to the developing embryo. The primary endosperm nucleus repeatedly divide to give rise to free nuclei stage. So this stage of development is called as free nuclear endosperm. Example for free nuclear endosperm is coconut water. Later on, around this free nuclei, cell wall formation occurs, so resulting into the formation of cellular endosperm. So here, the white kernel surrounding the coconut water is the example of cellular endosperm. Okay, so this endosperm, it may be present even in the mature seed. So if it is present in the mature seed, such seeds are called as albuminous seeds, or they are also known as endospermic seeds. Example here is castor and even the coconut, right? And in some seeds you may not see the endosperm because endosperm is fully consumed by developing embryo. So if the endosperm is absent in mature seeds, then such seeds are called as ex-albuminous seeds or non-endospermic seeds. Here the examples are pea, beans and then groundnuts. Now let us study about the development of embryo. Development of embryo from the zygote is called as embryogenesis, or it is also known as embryogeny. The embryo develops at the micropylar end of the embryo sac where the zygote is situated. Most zygotes divide only after the formation of endosperm. This is an adaptation to provide assured nutrition to the developing embryo. So here the zygote gives rise first to the proembryo, and then it gives rise to the globular stage, and then it gives rise to the heart-shaped stage, then it gives rise to the mature embryo. Let us now study about the structure of dicot embryo. A typical dicot embryo consisting of embryonal axis and two cotyledons. The portion of the embryonal axis above the level of cotyledon is called as epicotyle, and it contains plumule, that is shoot tip. The portion below the axis is called hypocotyle, and it contains a radicle, that is nothing but root tip. The root tip is covered by root cap. In monocot embryo, there is only one cotyledon, that is called as scutellum. The second one is actually highly reduced and it is called as epiblast. The embryonal axis below the level of scutellum is called as hypocotyle, and it contains a radicle, root cap, and these structures are enclosed in a coleorhiza. The region of the embryonal axis above the level of scutellum is called as epicotyle, and it contains shoot apex, leaf primordia, which is nothing but embryonic leaf, and these are covered by a hollow structure that is called as coleoptile. Okay, now let us see the development of seeds. Now seeds are the fertilized ovule which develop inside the ovary, That means they are present in the fruit, right? And these seeds are the last stage of sexual reproduction in angiosperms, that is flowering plants. Now, a seed consisting of seed coat, endosperm, and cotyledon. Now, in some seed, the endosperm may be present or it may be absent. So, based on the presence and absence of endosperm, the seeds are of two types. One of that type is albuminous seeds. Means in these seeds, endosperm is present. So, such seed examples include wheat and maize. In some seeds, endosperm is absent. Such seeds are called as non-albuminous seeds. Example here are pea, beans, and also groundnut. In some seeds such as black pepper, they have remnant of nucellus. Nucellus, we know that it is present inside the integument of ovule, which is a nutritive tissue surrounding the embryo sac. Right? So in some seeds, they have remnant of nucellus that is called as perisperm. Here you can see. So this is the example of black pepper. This one is an embryo. Inside that you can see the perisperm. What is that perisperm? It is the remnant of nucellus in mature seeds like black pepper. Clear? So moving to the next one. Let us now study the advantages of seeds to angiosperms. Pollination and fertilization is independent of water. Right? Seed formation in angiosperm make a reproductive process such as pollination and fertilization independent of water. Next, they have better strategies for dispersal. Here, seeds are having better strategies for dispersal to new habitats and help the species to colonize in the other areas. Nourishment by reserve food. Young seedlings are nourished by reserved food until they are capable of photosynthesis on their own. Protection to the young embryo. The hard seed coat provides protection to the young embryo. It creates variation. Being the products of sexual reproduction, they generate new genetic combinations leading to variations. The oldest seed is that of Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000-year-old viable seed is of the date palm, that is Phoenix dactylifera, discovered during the archaeological excavation at King's Herod Palace near the Dead Sea. Ficus, orchid fruits, and some parasitic species such as Orobanche and Striga contains thousands of tiny seeds. Now let us study about the development of fruits. The fruits are the fertilized ovary, right? The ovary is going to develop into fruit after fertilization. Ovary wall is going to become the fruit wall, that is called as pericarp. Peri means surrounding, carp means fruit, right? So here the fruits may be fleshy, as in case of mango and orange, or they may be dry, as in case of groundnut and mustard. Some fruits, they are called as false fruit because the fruits which develop from the part other than the ovary is called as false fruit. I repeat, the fruits which develop from the part other than the ovary are called as false fruits, right? So now when do we call it as a true fruit? If the ovary develops into a fruit, the fruit should be developed from the ovary, then only it is called as fruits, true fruits. If the fruits are developing from the parts other than the ovary, then they are considered as false fruits. So here the false fruit examples are apple and strawberry. In case of apple and strawberry, the thalamus is going to contribute in the formation of fruit. The part which we discard, that is actually the ovary. Surrounding to that is the thalamus, which is edible. And these are the examples of false fruit. Clear? Okay. Some fruits, they are developed without the fertilization. They are called as parthenocarpic fruits. Example here is banana. The fruits usually develops after the fertilization. The ovary is going to develop into fruit only after fertilization. But in some cases, like in banana, the ovary develops into fruits without fertilization. So such type of fruits are called as parthenocarpic fruits. Okay, now let's talk about the apomixis. Here apo means without, mixis means mixing. The process of formation of seeds without fertilization is called as apomixis. Seeds are the last stage of sexual reproduction. We know that sexual reproduction, where it involves the two gametes, fertilization of two gametes, then only the seeds are formed. The ovules are going to develop into seeds only after fertilization. But in some species of Asteraceae and grasses, the seeds are formed without fertilization, and such process is called as apomixis, right? And apomixis is a form of asexual reproduction mimicking the sexual reproduction. How it is mimicking? By producing the seeds. And why it is a type of asexual reproduction? Why it is a form of asexual reproduction? It is because there is no fertilization. There is no involvement of two gametes. Right? So because of that, apomixis is a form of asexual reproduction mimicking sexual reproduction. Right? As I told you, in some species of Asteraceae and grasses, the diploid egg cell is formed. Right? So here, how diploid egg cell is formed? Let us see the normal process. In normal embryo sac, we know that megaspore mother cell is present, which is diploid, which undergo meiosis. And after undergoing meiosis, megaspore tetrad, means 4 megaspores are generated. Out of that, 3 are going to degenerate, only 1 is going to become functional. From there, it undergo mitotic division, especially nuclear division, forming 7-celled 8-nucleated embryo sac that we studied in our previous video. If you have missed that video, go through my previous video and come back to this video. Anyhow, here the normal process is taking place where a haploid egg cell is produced. So after fertilization by the male gamete, which is a haploid, so two haploid cells, when they fuse together, forms the zygote. From there, the normal embryo is going to develop, which is diploid. But in case of some species of Asteraceae and grasses, here the megaspore mother cell, which is diploid, will not undergo meiosis. Right? So directly the megaspore mother cell, which is diploid, without undergoing meiosis, it directly undergo the mitotic division, only the nuclear division. So this diploid nucleus undergo mitotic division. So because of that, it is going to form an embryo sac where it is having diploid egg cell is produced. The embryo which develops here from this egg cell, it can be called as clone. So apomictic embryos are called as clones. Right? Now, apomixis is important for producing hybrid varieties of fruits and vegetables, and also for increasing the crop yield multifold. Okay, here there is one more condition that is called as polyembryony in some varieties of citrus and mango. Right? The seed contains many embryos, more than one embryo usually. So normal process, if you see, here there is a megaspore mother cell undergo meiosis, and it is going to form a normal embryo sac where it is having haploid egg cell. After getting fertilized, it will develop into a single embryo, right? But in some varieties of citrus and mango, what is going to happen? We know that surrounding the embryo sac there are nutritive tissue that is called as nucellus. So some of the nucellus cells, they divide and protrude into the embryo sac and develops into embryo. So in such case, ovule or the seed may bear more than one embryos. So after germination, you can see one seed is giving rise to multiple seedlings, right? With this, we have completed the chapter. Hope you found this video useful. If so, hit like button, share this video with your friends and classmates. If your teacher, share this video with your colleagues. I will see you in my next video. Till then, take care.